who have joined us this afternoon for this public lecture by Professor Giaca on the topic COVID-19, a journey from lung pathology to novel drug discovery. I'm G. Mahesh from the CSIR's Science Communication and Dissemination Directorate. As you can see, the DG CSIR is not with us right now as he's held up in an emergent meeting, and hopefully he will join us very soon. All the same, we are honored and privileged to have Dr. Ram Vishwakarma with us this afternoon. And we also have Dr. Chandrasekhar, Director, Indian Institute of Chemical Technology with us. Now, Dr. Vishwakarma is the advisor to the Director General CSIR, and he has been directing and coordinating the CSIR's COVID-19 efforts. Dr. Vishwakarma was the director of the CSIR Indian Institute of Integrative Medicine, and he has a keen interest and has also made significant contributions in the areas of drug discovery, medicinal chemistry, natural products chemistry, organic synthesis, chemical biology, and glycobiology. I will now request Dr. Ram Vishwakarma to give his opening remarks, introduce our eminent speaker, and steer the rest of the program this afternoon. Sir, over to you, Dr. Ram Vishwakarma.
Thank you, Mahesh. It's a special privilege and pleasure today that we have Professor Mauro Giaca with us. And in fact, thanks to virus, CSIR got in touch with him almost now close to maybe 10 months or more. And I think that journey of his phenomenal discoveries on virus and then translating that into a clinical candidate. So right now we are in the process of conducting phase 2 clinical trial, essentially based on his idea and his advice. So it's a special privilege. I welcome you, Mauro, to this talk. And I think Dr. Chandrasekhar made effort to actually make this lecture happen. We've been discussing in our CSG that, how do we sort of inspire our younger people who are into basic sciences, but if we give them examples of how this science which is done in the lab is actually translated to the bench, I think this was a good example, and that's why we requested Mauro to give this talk. Very briefly, I think he has a very, very distinguished career, so I will not sort of use his time trying to use my introduction, but very briefly for younger people. He's a professor of cardiovascular sciences at King's College London. He's one of those unique people, MD PhDs, which unfortunately in our countries are very, very rare. And that is, I allude, one of the fundamental reasons why we don't do clinical translation. So I think it's a special privilege. And until 2019, he was Director General of International Center of Genetic Engineering and Biotechnology. All of you know, there are 2 branches, and one in Trieste and one in New Delhi. And he was the overall DG of this institute, which is run by United Nations. And since 2005, he has been full professor of molecular biology at University of Trieste in Italy. Mauro is currently president of International Society of Heart Research, EU section. He is an elected fellow of Academy of Medical Sciences of UK, and obtains degree in medicine from University of Trieste, and PhD in microbiology, virology from University of Genoa, Italy. Had a very distinguished, accomplished career, and any contribution in virology, in molecular biology, and of course their clinical translation, over 370 plus original publications, and very recent one that we saw, I think, 10, 15 days ago in Nature, which was actually the work which led to the clinical trials on the glucosamide in India, and where he actually identified the very important new mechanism of damage in lung, and the protein, basically the calcium-dependent scramblase, a protein which is mainly responsible for exposure of PS, which is a sort of a death signal, and how he figured out using all of these autopsies, and he kept telling us in the beginning that how those autopsies of people who died of COVID gave these clues that how this lung damage is happening. So that mechanism, and then he screened close to 3,000 approved drugs. And out of that screening and the work, this very important anti-helminthic drug called niclosamide came right on the top. And that mechanism then, became the foundation of translation. And simultaneously, after his idea, we also started working on this separately, and we recently have sort of, along with Dr. Satyajit Mayer, we have published this study on niclosamide, which is further adding that actually it inhibits the viral entry and sort of neutralizes the endosomal pH. So I think this is a very threefold mechanism for a very small molecule, and I think that story everybody is looking forward to hear. So I will not take much time. Welcome you, Mauro, for this talk, and the floor is yours. Thank you very much.
So thank you really very much for the opportunity to be here. Thank you, Dr. Vishwakarma, Dr. Chandrasekhar, and thank you to Shekhar for the opportunity and for the splendid interaction over the last 10 months or so. So the story I'm going to tell today is the story of this pandemic that has hit so hard the entire world and is continuing to hit several countries, including I'm afraid India at this moment. And so, but best wishes really for the country to get out of the acute problems at least as soon as possible. So something that became absolutely clear is that we were not ready for this pandemic in the entire world. This is a cover of Time, inspired by the Ebola epidemics in Africa, and the title was quite telling. That was 2017, so well before SARS-CoV-2 appeared. That is, we are not ready for the next pandemic, and we proved ourselves not to be ready. And this is an important pandemic, which can be seen in this infographics, in which the size of each of the balls represent the importance of the pandemic in the history of humanity in terms of death. So these big balls here are AIDS, who took a toll of 35 million people worldwide and is still around, or the flu pandemic, which took a toll of 50 million people. To go back with the bubonic plague, so the Black Death, 200 million people dead, and this is the size. And COVID-19 is here, so it is, not irrelevant in the history of human infectious diseases. And as you know, everything started at the beginning of 2020 with these communications at the Chinese CDCs of the appearance of cases due to the presence of a novel coronavirus. So the virus probably was there several months in advance before it was detected, and the first publication on patients was at the end of January 2020 with the description of the clinical features of 99 cases of this new pneumonia. I'm showing these slides because we tend to think that COVID-19 is a virus that infects lung cells and kills lung cells, which is obviously the case. But the most remarkable feature of this virus is that it has clinically determines a number of features which are quite unique, and to say the truth, for most of which we don't have still a reason. For example, it is unclear why men are more affected than women, or why people who are obese have a greater risk of being severely diseased, why it has different prevalence in ethnic groups. The COVID-19 hit first Chinese people, and then it moved to Europe, to Italy, where there is not ethnic variability. But then when it started hitting France and the UK and the US, it was immediately clear that Black people are more affected, but there is no explanation in scientific or medical terms for that. What is most relevant, it is absolutely unclear why older people are more at risk of contracting a very severe disease compared to young people. A young boy has a chance of getting severely diseased of 1 out of 500,000, which means extremely rare, compared to an individual who is more than 80, who has a chance of having a severe disease of 1 out of 8. And the reason for this difference is completely unknown. So why there is this age dependency? Then there are some symptoms that are quite peculiar. For example, one of these is Hepa Hypoxia. So people who get the disease, they seem to cope quite well with that. But then somebody measures the amount of oxygen in the blood, so what's called oxygen tension in the blood, and it is very severely reduced without the person appreciating that. That's called the conditions of Hepa Hypoxia, and some people in these conditions, from being well and going with their legs to the hospital, then find themselves in intensive care units in the afternoon. It is completely unclear why there is a clotting of blood vessels in the lungs, which is very specific for these conditions. And then there are other symptoms. So there is diarrhea in 20% of the patients. The patients in intensive care units have intense sweat, and a large number of people who are hit by the disease, both severe or almost asymptomatic, has as a symptom the loss of sense of smell and taste. So clearly there is something we don't understand of this disease, and something that requires a mechanism that is still lacking. So these clinical features became almost immediately apparent, and so we started thinking that, well, what we don't know of this disease is what really happens of people who contract the infections. And unfortunately, especially at the beginning of the pandemic, the number of people who were admitted to hospital and died were quite high. There's been a worldwide in absence of information for several months of what was the post-mortem analysis of these of the individual who died of COVID-19. There was fear of performing these analyses. An exception to this was the pathology department of the University of Trieste, Italy. And this is the same place where, as Dr. Vishwakarma mentioned before, it's the same place where the International Center for Genetic Engineering and Biotechnology is located in one of these two seats. You know, the other seats, which is even bigger, is in New Delhi in India, and works in close collaboration with the Department of Biotechnology of the government of India. And so we took advantage of the availability of post-mortem samples from patients who died of COVID-19 to perform some of the first analysis of the organs of these individuals. And so we started analyzing 41 patients, consecutive patients who died. And what we found was part expected from the clinical and the radiological characteristics, and part largely unexpected. What was expected, that the damage to the lungs was really very severe. The structure of the lungs was almost completely destroyed in some patients, with a large number of inflammatory infiltrates, and the substitution of the lung tissue with scar tissue, so formation of fibrosis in the lung. This is very important. So the virus really leads to a massive destruction in some patients of the lung structure. You have to consider that these patients are patients who actually died of of the infections, and which infection was really severe. That was somehow expected. What was less expected was the extent of thrombosis, so coagulation of the blood in small and large arteries. These are some of the first pictures last year of large thrombi, so blood clots that block even the large pulmonary vessels. As you see, there's lungs that are completely infarcted because of the presence of these thrombi blocking the circulation. And this phenomenon was evident in almost 90% of the patients we analyzed at the level of the small arteries and veins, or the microcirculation. These are images of lungs, so one could even detect that these are lungs. So the air structure is completely destroyed, substituted by this sort of scar tissue with a lot of inflammatory cells, and all these red structures are blood vessels which are blocked by a clot. So this microthrombosis, which was very, very evident. And what was also surprising us is that these blood clots were different in age. So some of these were very fresh, as if they had occurred 1-2 days before death. But some of them were already converted into fibrous tissues, meaning that they occurred weeks before, which means that this tendency of the blood to coagulate or form thrombi in the lungs is a property intrinsic to the lungs, because you can't find this in any other district in the body. But what was also evident that there was a lot of virus. This in blue is the presence of the virus seen with a probe that sees the viral genome inside the lungs, and this virus was often present in cells which had a very abnormal structure. You can see here large cells, often with 2 nuclei, these structures here, on a very large cytoplasm outside the nuclei. And these are examples of other cells. Here they are seen with an antibody that recognizes the surface protein of the virus. It's called the spike protein. You see these very abnormal cells which are infected by the virus, persisting for 30-40 days after infection and until the patient dies. The most surprising characteristic, however, something we completely did not expect, was the presence of cells with very abnormal cytological features, and in particular very large cells like these ones, containing several nuclei, sometimes up to 20 different nuclei. And these multinucleated cells are known in biology with the name of syncytia. So these are the product of fusion of different cells. So 2, 3, 4, 5, up to 10 cells that fuse, or 20 cells that fuse, form these large syncytia. And these syncytia were cells derived from the fusion of typical lung cells, what we call in biology pneumocytes. And so clearly they were a product of something occurring at the level of the lung cells. These are examples of other syncytia. These are other examples of these cells. This is a syncytium formed by the fusion of the 2 large cells. Here there are probably 15 different nuclei inside the cells. You can see other pictures, and these were present in the vast majority of patients to different extents. You see large syncytia in the lung, close to 2 vessels down here, which have clots, so they have thrombi in their internal part. You see there's a close-up of this large abnormal syncytia. When we published this paper in spring last year, there was a big rumor here in England and also elsewhere, because that was the first demonstration that SARS-CoV-2 is really a virus that damages severely the lungs, so in which the picture you can see in the lungs is a picture of the strongly, massively affected organ. There was much scare of that also in the media, because obviously this means that also people who recover from the infection and survive have the problem of coping with this type of destruction of part of the lung structure, which might be responsible for what is known as at least in part of the long COVID. So basically this prolongation of symptoms, this difficulty in breathing, difficulty in exercising, that persists for several months after infection. And so the story from Trieste moved back to the King's College London. This that you see also in the back of my screen is the James Black Center. I forgot to say that we started this work as an emergency research supported by the university here at the beginning of the pandemic. But the reason why what we do here in the laboratory is cardiovascular work. So our specialty is how to use gene therapy vectors, so viral vectors, to induce regeneration of the heart after myocardial infarction. In England, you see immediately when the pandemic started, there have been a recruitment of all scientists who would give a contribution to work on COVID-19, so to switch to work on COVID-19 to seek all kind of expertise in trying to understand this disease, and there were funds available to do this research. So since we had this expertise in virology coming from the gene therapy field, we reconverted our activity to work on COVID-19. And so we started reasoning that we wanted to understand why there was this big syncytia. What is the mechanism by which the virus can cause this syncytia? And the most likely explanation at the time, which turned out to be true, is that this is a property of the spike protein of the virus. So the viral particles of SARS-CoV-2 have a membrane outside. This is a membrane that comes out of the cells where the virus is produced. And on the membrane, there is a protein that's called spike. And this is a protein that the virion uses to bind to its receptor. And this receptor is, in the case of SARS-CoV-2, is the ACE2 receptor. After binding, spike is activated, and the property of spike is to induce fusion of the membrane of the virus and the membrane of the cell that the virus infects. So we started thinking that it was the presence of this spike protein that was responsible also the fusion of different cells, and so we obtained a way of expressing the spike protein into the cells, and then we tested whether the cells can fuse as a response to spike. And what we saw, and that was around March, April last year, was really remarkable. And this is our first experiment in which basically we took cells here on the left. These are normal cells that express this ACE2 receptor. The boundary of the cell is in red. The nuclei of the cells are in white. And expressed into these cells, the spike protein of this coronavirus. And you see that just after overnight, we saw the formation of these enormous syncytia. Here the spike protein is colored in green. You see these large lakes of cells, so fused cells with several nuclei in the middle of the cells, and which are the product of the fusogenic activity induced by the spike protein, so the capacity of fusing cells. I confess that when I first saw this experiment, I mean, I was even scared, because you have to think of this type of events occurring in the lungs of an infected individual. And this is due to the fact that the SARS-CoV-2 has this intrinsic capacity of fusing membranes. And when a cell is infected by the virus, a part of the spike protein is expressed on the surface of the cell, and through this spike protein, then these infected cells can fuse to neighboring cells, forming these very large syncytia. Just to give you an idea of these events, this is a movie in which we express the spike protein. Here it is in green. And I hope you can see in my movie, but you see that just in 5 hours, these cells move around, and when they find neighboring cells, they start fusing, and they form these very large syncytia. These are other fields in these 5 hours cultures in which you see the formation of these very large syncytia. So there is a property that is specific for this coronavirus, that is to fuse cells in response to ACE2. For example, the spike protein of the SARS virus, who was the first coronavirus to cause a small pandemic, 800 people in 2003, 2004, that fortunately then got extinguished. Well, this spike protein doesn't have this fusogenic capacity. And these are very nice pictures of these syncytia obtained in the laboratory. Here you see the nuclei in blue, the spike protein is in green, and the cell skeleton, the cell architecture is in red. So these are very pathological cell characteristics, some other syncytia. And you see that these cells emit these prolongations to which they start contacting other cells. These are normal cells in red, and this is a cell expressing spike, so they change shape, they start emitting these. In biology they're called philopodia, and these contact neighboring cells, searching for the receptor and almost trying to convince them to fuse. You see these infected cells that express spike, and this spike protein emanates contacts. So this is where we started to think, well, perhaps COVID-19 is not a disease in which there is a virus that enters the cell and replicates, kills the cells, and that's gone, which happens with several other infections. For example, most of the pathological effect of influenza are due to killing of the cells. It's not even a virus in which most of the pathological effect are due to the hyperinflammation it causes. Again, in the case of influenza, there is an hyperinflammatory response. In some cases, is responsible of disease. Perhaps COVID-19 instead is a disease in which there is a strong third component besides killing of cells by the virus or inflammation. The third component is the presence of cells that fuse with other cells and exert pathological effect. So the capacity of the infected cells to exert pathological effect. Something we have here in the Department of Cardiovascular Sciences is a robotic instrumentation, like which is the one that you see. And this is a full robotic cabinet, completely automated, in which we can perform robotic screenings. And we have several libraries, collections of genetic compounds and chemical drugs that we can screen. And in particular, we have two collections or two libraries of drugs that have been already approved by the FDA in the US or the EMA here in Europe for other purposes. And collectively, these drugs amount to 3,800 different compounds, approximately 3,000 different drugs. Some of the drugs are overlapping between the two libraries. And so we asked ourselves, well, let's see if we find some libraries, some drugs from these collections that block the formation of these syncytia. Here the assay was very simple. So we expressed the spike protein inside the cells fused, and at the same time we screened for drugs that block this fusion. These are again some nice pictures of fused cells in the absence of the drugs. And these are examples instead of a very successful drug that permitted the spike protein to be expressed. Some cells are still green, but they don't fuse to the other cells. So this drug blocks completely the fusogenic capacity of the cells. And here on the left, you see the cumulative results of all 3,800 drugs. So each dot is the effect of different drugs. If the effect is 0, it means the drug doesn't do anything. If it is negative, the drug is very, very effective. And those that are below in this scale are really very statistically significant effective. So there are 5 standard deviations below the 0, so below the mean. And the most effective, and we run 2 different screens with 2 slightly different assays, and in both screenings, one of the most effective drugs was in blocking synesthesia, was called niclosamide. It was the first in one screening and the second in the other screening. Altogether, these 2 screenings revealed 83 drugs that were very highly statistically significant, those in red here, at blocking formation of this synesthesia. So out of these 83, we shortlisted them for pharmacological reasons to 43, and then we teamed up with a team of virologists led by Wendy Barclay, who's also part of the committee that informed the government here in the UK, and to test them for cell protection against SARS-CoV-2. The assay was like this one. So basically here you see these strips are cells in the presence of different concentration of the drugs. You see this is a control, so cells survive. If you put the virus, the cells die because the virus kills the cells. But if you put the virus in the presence of the drugs, the cells keep surviving. Look at the niclosamide. Basically, the cell survive even at very low concentration of niclosamide. So it's very effective at blocking this pathological effect. And as a consequence, it's also very effective at blocking viral replication. This is an experiment performed with another virology group here at King's College London, led by Michael Mullin, who is a huge expert in several viruses, starting from HIV. And you see that basically there is a dose-dependent capacity of niclosamide of blocking viral replication with a concentration, effective concentration, which is well within the range that can be used therapeutically. Another drug that came always positive in our assay is called clophazamine. Niclosamide, you will see in a moment, is a drug that is used to treat worm infection. Clophazamine is the main drug that you use against leprosy. Is the drug is most effective against the bacterium that cause leprosy. Obviously, we were interested in understanding how can these drugs block the formation of these synesthesia, and we were back a bit taken aback by the fact that the 83 drugs that were effective belong to very different classes and mechanism of action. And so we started studying these drugs as classes. And what we found was that, you see here, for example, there were, these are the different pharmacological classes of the drugs. And these are their effect in the screening. 0 means non-effect. Here on the left are the drugs that are more effective. And you see immediately that there are classes that are very, very effective. So, for example, this group of drugs here is very effective, and this group of the drugs here, and we were surprised by, for example, by the fact that among the most effective drugs there were 5 antihistaminic receptor drugs, 11 antipsychotics, 8 antidepressants, and these drugs have nothing in common except one feature, that they all reduce the level of calcium ions inside the cytoplasm. So all these drugs, so histamine anti-receptor antagonist, antipsychotic, antidepressant, work by blocking the effect of a receptor that's on the cell membrane, with the main function of which is to induce the release of calcium from the stores inside the cells into the cytoplasm. So basically these drugs block the levels of calcium in the cytoplasm, which tended to indicate that there is an involvement of calcium in the formation of these synesthesia. So we teamed up again with another group of electrophysiologists here at King's College London. Juan Burrone is the person who leads this group. And here this is an experiment that we perform with them by having a cell expressing an indicator for calcium. This is a fluorescent protein that if calcium is not present is black. If calcium is present, it becomes green. This report is called GCAMP6. And here we are culturing these cells together with cells that have spike protein on their cell membrane, and these are in red. So basically, the red cells have spike, and these cells have a sensor for calcium. And see what happens. You leave these cells growing together for a few hours, and they start fusing, and every time they fuse, there is a tremendous release of calcium inside these cells. So these fused cells, start flashing in response to this increase of calcium. So basically, calcium is required for the formation of these large syncytia. This is an overnight observation, accelerated, and you see that it seems to watch a Christmas tree lightning, because the cells, as they fuse, they start having these oscillations of calcium in their cytoplasm. So we found that there was a connection between formation of this cell fusion and calcium, and this can be quantified. You see, these are these spikes of calcium, so this oscillation of calcium. If you give the drugs, in particular niclosamide or clophazamine, you completely blunt this activity. So the drug work because they block calcium. This is an example. So you see the Christmas tree in the control, or this drug, which is not much effective, but the Christmas tree is completely abolished in the culture where you have niclosamide or clophazamine. So there is a connection between calcium and the formation of these syncytia. So the more calcium is released from the stores inside the cells, the more syncytia there is. But how does this work? And here, what we observed is that there is a category of protein, a class of proteins, which are very sensitive to the level of calcium and are activated by calcium. And this is a family of proteins which are called anoctamines or TMEM16 proteins, which are activated by calcium. And when they are activated, they do two activities. One is they let chloride ions to go outside the cells. And the second, they change the composition of the cell membrane. So they let a special lipid that is always on the internal part of the membrane to go to the outside part of the membrane. This lipid is called phosphatidylserine. And one of these proteins, in particular TMEM16F, this is a family of 10 different proteins, it is expressed in all cells, including pulmonary cells. And what is interesting is that if you look at what is known, it is these TMEM16 proteins are essential for all biological events that are involved in cell fusion. So for example, cell fusion occurs in the formation of the placenta. So syncytial trophoblast forms, so big, large cells with many nuclei, because this TMEM16F protein is present. Mice in which this is the gene for this protein is knocked out, they don't fuse. Or sperm cells during fertilization expose phosphatidylserine. Or when satellite cells fuse to become muscle fibers, again, these proteins are required. So all events that require cell fusion require this protein. So we rushed back to our experiments and we inhibited the formation of 16F and we completely blocked the capacity of the cells to expose this phosphatidylserine. So it is true that also in the cell type that the virus infects, phosphatidylserine, the exposure on the outer part of the membrane requires these proteins. And all the big syncytia express this phosphatidylserine on the outer part of the cells. So these are normal cell culture. These are culture expressing spike. they form syncytia. Here the nuclei of syncytia are shown using a marker that stains the nuclei. And you see that in green we can see phosphatidylserine, and these syncytia have plenty of phosphatidylserine on their outer membrane. So you have to think that these syncytia form, and they have a lot of these phosphatidylserine on the outer membrane. We rushed back again to patients, and we found that all syncytia in patients are enriched in phosphatidylserine. Here in gold, in brown, we can see phosphatidylserine on the outer membrane of these syncytia. So basically the mechanism now becomes enriched because we have calcium that is activated. This calcium activates these channels on the membrane, and these channels permit the externalization of phosphatidylserine or the outer part of the membrane. What happens if you block this protein, so this TMEM16F? Well, you block completely syncytia. You see that if you remove the receptor or you remove the TMEM16F, in both cases, these large syncytia are completely, almost completely abolished. And the drugs do this, in particular niclosamide and clophazamine. So they block externalization of phosphatidylserine, and in particular, niclosamide is a direct inhibitor of these TMEM16 proteins. Clophazamine, we don't know how it works, but we tend to suspect, and we are investigating this in these days, we tend to suspect that it might also block directly the channel. And so basically what we found in our screening, what our screening revealed, is basically that blocking syncytia is a property of drugs like niclosamide or other drugs of the same family that are direct inhibitors of these channels. Clophazamine perhaps could do the same, or a class of drugs that by other means diminish the level of calcium, which in turn doesn't activate these channels. And this is a case of the histamine 1 receptor antagonists, the anticholinergics, the antipsychotics, and so on. Basically, so this is how from the screening we understood the mechanism. And the mechanism probably tells us also something on the pathogenesis of COVID-19. So this is again the list of the many unknowns. And we believe that some of these unknowns could be explained exactly by this mechanism. For example, if we give spike to the cells, there is increased secretion of chloride. This is seen by measuring currents induced by chloride. And if you block 16F or you give niclosamide, in both cases, you completely inhibit this. And secreting chloride is the mechanism that could explain diarrhea, or it could explain Hepa Hypoxia, because Hepa Hypoxia is a mechanism by which basically you rapid deterioration of lung function could be due to secretion of water. And what chloride does when it is secreted, it attracts water. This explains diarrhea. It can explain edema, so filling of the lungs, also filling of the lungs with water. What is even more interesting is that activation of this TMEM16F is a main mechanism that activates platelets. So platelets are known to be first activated and then to aggregate, but then when they have to become pro-coagulogenic, so to induce formation of blood clots, they use phosphatidylserine secretion for this. And this increases up to 10,000 times the capacity of function of platelets, and so formation of clots. And indeed, if you take a virion that expresses spike, you give it to platelets, they get activated. Or if you have cells that express spike on the surface, they attract platelets and platelets aggregate onto this. And we are convinced now that this might be a main mechanism that induces pro-coagulation activity. So it makes the lungs, which are infected, the perfect organ in which the coagulation system get activated, and this explains thrombosis in the lungs. And then eventually, this could explain also loss of smell and taste. You might remember that as far as smell is concerned, there are olfactory cells present in the upper cavity of the nose. So this is the nose, very high in the nose. There are these cells, and these are neuronal cells then that emit an axon that crosses the bone and reaches a part of the brain which is in the frontal cortex, which is the first station that drives the signal, electrical signals that drives the sense of smell to the brain. Well, these cells in the olfactory epithelium, they are infected by the virus. This is again post-mortem analysis of patients with COVID-19, in which we stained the olfactory epithelium, so the upper part of the nose, where this special structure exists, with antibodies recognizing the nucleocapsid protein of the virus or the spike protein of the virus. And all these in brown are infected cells. And it is known that these cells use TMEM16A, TMEM16F to transduce the electric signal. And we can also confirm that if we stain the olfactory epithelium for TMEM16A or TMEM16F, there are these cells express, different types of cells in this olfactory epithelium express these proteins. So we believe that the disturbance in the sense of smell and the taste goes more or less on the same way, is due to the fact that the virus infects these cells and perturbs and activates these TMEM16 proteins, and this leads to the loss of perception of smell and taste. Obviously, given this, niclosamide becomes even more interesting, not only as an antiviral agent, so an agent capable of blocking viral replication, and Dr. Vishwakarma has reminded us of a beautiful work performed in India, in Bangalore, on the capacity of niclosamide of blocking internalization of the virus, but perhaps also, and perhaps mostly, as a factor capable to, as a drug capable to block the pathological effect of the infected cells on the body, and in particular thrombosis and the other damaging events. Niclosamide is a drug that was developed in the 90s. It's a synthetic drug, doesn't exist in nature. It was originally discovered as a drug against nails, and then in 1982 it was approved to treat tapeworm infections, so infection from taenia in the gut, and it is in the list of the WHO list of essential medicines. Is very effective against the worm infection, relatively inexpensive, and there are now more than 20 clinical trials for niclosamide, several of which have started, especially after our work was published for COVID-19. The most advanced clinical trial is here in India, or there in India, I would say, I'm in London in this moment, which is called NICOVID, which is a randomized phase 2 clinical trial that is ongoing through the auspices of CSIR and the interest of the Laxai Life Science company. This trial is recruiting at this moment, and in a couple of months we should be able to see whether niclosamide is effective. The primary endpoint of the trial is the capacity of niclosamide to block progression of the disease. It is in hospitalized patients to see whether treatment really is capable of blocking the pathological effect of the virus, and only as a secondary endpoint is the capacity of clearing out the virus. We are really very anxious to see how this trial will go, and that with this proposition and concept notion that the COVID-19 is essentially due not only to the virus killing the cells, but to the pathological effect induced, will hold true. All this work was performed as a collaboration with by very talented investigators, a group of a few people that who during the entire lockdown work night and days and weekends to do the screening and characterize the effect here at King's College London, in collaboration with ICGB and the University of Trieste for the pathological part, and with the essential involvement of Wendy Berkeley and his group, Mike Mullin for the virological part, and with Juan Burrone for the part related to electrophysiology and chloride activation. And that was my last slide. I thank you very much for your kind attention, interest, and the invitation. Very happy to take questions if time permits, and there are any. Thank you very much.
Thank you. Mahesh, how you have organized the questions?
Sir, we can start interactions. So as questions come up in the social media, I will take it up here.
So you will receive them and then you will.
Yes, I'll keep looking at it. Yes, sir.
Yeah. Okay. Oh, this is a fascinating presentation. I reading paper was one thing, listening to you is another thing. That's amazing. You know, he says the work that started in Trieste Hospital, and then how it came to King's College, and it's amazing. I think it's a very, very interesting, and I think a lot of new biology that came out of this work beyond niclosamide and activities of these drugs. I think more fascinating is the role of basically the calcium and the role of the PS, and then fusinic activity of this. So I think if I can ask you that among all the kind of spike protein which are there on these SARS, MERS, and all these viruses, it's basically there's a common strategy they talk. It is some special property that SARS-CoV-2 has acquired about the fusinic activity, or other viruses in the same, they also have.
That's a very interesting question. I mean, there are several other viruses that induce fusion of cells. The most famous one is the respiratory syncytial virus. However, the mechanisms are different. And in terms of coronaviruses, the MERS virus is also fusogenic, but it binds to a different receptor than ACE2. And we don't have information about MERS and lung thrombosis because there are no autopsies for religious reasons, being the infection of MERS in the Middle East. Basically, there are no autopsies where we can look at that, but the receptor used is different. The SARS virus is definitely not thrombogenic and doesn't induce fusion and doesn't activate this mechanism. The difference between MERS, SARS-CoV-2, and SARS is the presence of an additional amino acids in the spike protein, which permit activation of spike at the plasma membrane level. So while the SARS virus, activation of spike means that the spike protein needs to be cleaved to become active. And for the SARS virus, this occurs only when the virus has entered in the cell by endocytosis, because it requires an acidic pH. While for SARS-CoV-2, the addition of these amino acids permits activation by proteases that are present in normal conditions, of furin proteases. And so activation can occur at the plasma membrane level. So this means that a cell infected by the SARS doesn't fuse. A cell is infected by SARS-CoV-2, then it fuses.
Oh, yeah.
So this is what we believe that is quite special. So this furin site is I mean, a very important feature of this virus.
Yeah. So then the main reason for the sort of acquisition of fusogenic activity by the spike proteins are obviously these glycans which are decorated, 50 or whatever sites are there. And as we see during the different variants that are coming out, Delta variant or Delta plus variant, there is a reshuffling of these glycans onto spike proteins, because most of the amino acids that are being shuffled are the one which are basically uses these N-glycan O glycan sites.
Yeah.
So
we have data now showing in which we compared the fusogenic activity of the different spike variants. And we can see that there is a broad correlation between infectivity, let's say, of the different variants and the fusogenic capacity. So let's say all the variants fuse, but for example, the Brazilian and the South African variants fuse more than the British variant or the European variant. And so meaning that all the variants maintain this furing site, cleavage site, and so they are fusogenic. And then the capacity to fuse can depend, for example, on the glycans and so the affinity with the receptor. But the fusogenic capacity is already there.
Yeah. So I think the main recent mutation that was reported, additional mutation, which is in the Delta plus, where there is a very close to furin that site. I think that's where this has required a much greater, I think 60% more sort of infectivity, or So I think that means then this study can then further be sort of looked into the pathology caused by the different variants. I think this was the original DG variant that, the first one which came out, the Wuhan variant.
The one we used is not even the DG, which was in Europe, but the original Wuhan. So we cloned the Wuhan virus. All the synthesia I show are the Wuhan virus.
Yeah. Okay. Yeah. Great. Yeah. Chandra.
So I think Mahesh has some questions. Mahesh, over to you.
Yes, there are a couple of questions on Facebook. Couple of questions are from Yogesh Taparia, who asks, can we expedite the use of niclosamide in patients? And at what stage or severity of the infection do we expect this drug to be effective?
Well, I mean, the testing drugs always require proper clinical experimentation. And so, I mean, one shouldn't rush using drugs in an appropriate manner. Clinical experimentation is ongoing in different centers in India, and so we just need to wait and see the results. Then the issue, if it works, will be that of speeding up. My impression is that, my opinion is that this could become a perfect drug to block the evolution of the disease. So to impede that the disease from being mild becomes severe. Some bodies are thinking that it could be also preventive of infection in earlier phases, of more severe infection in earlier phases, but I think that only clinical experimentation can tell that.
Thank you, Professor Moro. There are two more questions. This is from Nikhil Patak, who asks, what is the expected potential of niclosamide in combination with direct-acting antivirals like remdesivir? Are there any combination studies that are going on?
No, at least not as far as I know at this moment. But if the paradigm that niclosamide works on the pathological effect while antiviral work on viral replication itself is true, then I would see a combination as perfect to target the disease.
There's one more. This is from Arun Sharma. So he says, very nice presentation. And the question that he asks is, what I understood, he says, from your PPT, what I understood was that calcium-rich diet can be destructive use during COVID-19 infection. Am I right?
No, unfortunately not. I mean, because calcium is absolutely fundamental for the cells to a point that it is the cells that control their levels. And so we cannot influence the intracellular levels by food, unfortunately. So there is no relationship between what's inside the cells and how we can alter these levels by food intake.
Thank you, Professor Mora. These are the questions for now. Over to Dr. Chandrasekhar.
Yeah, thanks, Mahesh, for taking care of the event today. Professor Moro, thank you so much.
Thank you.
A wonderful talk. I think as Ram told, while we read your paper very, very closely and critically and started working with you, but I think the way you presented has been phenomenal. And I think many young minds are excited to be part of our teams to see how best they could become future Moros, hopefully.
That's very generous. Thank you very much.
And of course, you are going to be a role model hero for the Indian population. Ramya, over to you. Sorry.
Yeah, just to stop you, because I could not sort of stop myself asking this. So, Moro, what I am thinking right now, starting niclosamide trial, we also started, as we told you, colchicine trial, which is in very advanced. In fact, we have much larger recruitment. And interestingly, you'll be surprised, although scientifically it is, not mature, but many doctors in India actually have started using as an off-label use niclosamide and colchicine. So because of that, I think when the quality scientific information came about, so I think because this is a very safe drug and doctors are allowed to use as an off-label. So that's one part. But the second part, Chandra and I have been discussing, and we will take your input, that I foresee like a antiretroviral sort of cocktail situation which emerged for HIV, which is now state of the treatment. I foresee that situation in the future for this virus also. And this is not me, everybody, I'm sure people who know, they will be thinking about that. So I am trying to do a clinical trial, planning a clinical trial where we put 4 drugs together. And obviously niclosamide is one of them. And one of the drugs that we are thinking about, which recently we are interacting with the Spanish group about the tenofovir, right? This is the part of this prophylactic HIV combination which is available right now, once a single day. So what sometime maybe later, based on provoked by this presentation, I think we have a good opportunity to really scientifically, rationally sort of put together a combination of 4 drugs. I was thinking, Chandra and I have been discussing that we can initiate a trial where we combine the mechanisms, safer drug, keeping in the pharmacokinetics and all that. And is it possible to do that? So maybe we will, maybe shortly, we will take your help in this direction, and I will.
Yeah, well, I think it makes a lot of sense, because I think that in the future we will probably have a SARS-CoV-2 specific drug, for example, a new inhibitor of the protease or the polymerase developed just for this. But for the time being, when we have to do repurposing of drugs, mixing them together could be, with a rationale, could really be a way to have effect.
Yeah. So we will then take that thought from this presentation, and then maybe we'll come back to you again, take your input. Yeah, Chandra, over to you. Yeah.
Pleasure.
Sorry.
Thanks, Ram. I think, Morgan, I think on behalf of CSIR, Dr. Shekhar Mande, we are very, very delighted and to listen to your talk. And thank you so much for being with us. Of course, your talk, while has mentored and motivated many young colleagues, we also believe that hopefully once our clinical trial is done under your mentorship in partnership with Laxai, hopefully we would have the cheapest care for COVID-19, because this drug is available at almost like $10 per kilo. So hopefully we'll have the cheapest drug possible. You've also shown the power of collaborations. I think when your lecture was going through, the kind of collaborations you have built within your King's College and also, of course, in Italy with your collaborators, the way every point, every slide has the smell of a collaboration. So this also tells a message to our research community that I think not to work in silos, but to partner with collaborators to take science forward. And we do believe that our clinical trial data would be out, and hopefully we will handle the predicted third wave better with repertoire of repurposed drugs. And as Ram mentioned, certainly the colchicine, the niclosamide. You also spoke on clophazamine, which I think we did not take up, but certainly a small trial, clophazamine, taking some cue from your data, hopefully, I think should us take forward. Again, I think on behalf of DGCSIR and entire audience who are on the YouTube and Facebook, they have no access to talk to you directly. So I thank on behalf of all those viewers and listeners. So thank you so much, and looking forward to collaborating with you further. Thank you, Mauro.
Well, with great pleasure, and thank you very much for the kind words and interest. I only can say that it has been a pleasure interacting with you over the last several months, and I hope that we continue further. So thank you.
Thanks, Mauro. Thanks, Ram, and thanks, Mahesh. We are signing off for the day. Thank you.